This video lecture is on Chapter 1, which is Introduction and Cellular Structure. What is biochemistry? Biochemistry is the sum of chemical processes that occur in living organisms. So the way that I like to think about it is these chemical reactions is what really drives biological processes. Biochemistry involves understanding the levels of structural organization of biomolecules that are put together to make a cell. Small molecules such as nucleotides, amino acids, and sugars are single or monomeric units. These monomers can be linked together into larger biological molecules called macromolecules such as DNA, protein, and cellulose. The macromolecules can further be assembled into larger complexes that serve important structural and functional roles in a cell, such as forming the cell wall, the plasma membrane, or chromatin, and ultimately come together to make a cell and its organelles. There are 4 major types of biomolecules that are the constituents of cells. These are nucleic acids, such as DNA, lipids, proteins, and carbohydrates, such as glucose. Monomers are linked together to make polymers. Monomer nucleotides combine by polymerization reactions to form nucleic acids. Amino acids combine by polymerization to form proteins. And monomers of sugars can combine by polymerization to form polysaccharides. The process of polymerization occurs via dehydration reactions. For example, to link 2 sugar monomers together to make a polysaccharide, a molecule of water must be removed between them. Biological macromolecules and their building blocks have a sense or directionality. So what that means is the chemical nature of the ends of biomolecules are different in composition. This occurs at the monomer level and gives rise to polymers with different ends. And the chemistry of polymerization occurs in a specific orientation. In addition, the order in which monomers are assembled into a polymer also matters. In particular, the orders of monomers in proteins or nucleic acids can have different outcomes. So, for example, let's take the 4 letters A, M, D, E. These letters can be combined into different ways to make different words, such as dame, maid, and mead. And while these words all have the same letters, they all have different meanings. So macromolecules work the same way. There are 2 major types of organisms, which are prokaryotes and eukaryotes. Prokaryotic and eukaryotic cells have cellular features in common that are vital to life, such as a plasma membrane and ribosomes. Eukaryotic cells have additional features such as organelles and a nucleus, which makes them more complex compared to prokaryotic cells. Prokaryotic cells are the type of organisms living today and probably most resemble the earliest type of cells that existed as life evolved on Earth. Prokaryotic cells lack a membrane-bound nucleus, so the word prokaryote literally means before the nucleus. Prokaryotic organisms include bacteria and cyanobacteria, and these are actually very small single-celled organisms ranging from 1 to 3 microns in diameter. So a micron is the same as 1 micrometer. Again, while prokaryotes are single-celled organisms, they actually display group behaviors. So, for example, groups of them can exist in association to form colonies. Eukaryotes, the word means true nucleus. Eukaryotes contain a well-defined nucleus that holds genetic material that is set off from the rest of the cell by a membrane. Having a nucleus is one of the chief features distinguishing a eukaryotic cell from a prokaryotic cell. Eukaryotes can be multicellular. For example, animals and plants are eukaryotes. Any multicellular organism is a eukaryotic organism. Eukaryotes can also be single-celled. So yeast and paramecium are single-celled eukaryotic organisms. Eukaryotic cells are more complex than prokaryotic cells, and they're also usually much larger than prokaryotic cells. So they range from 10 to 100 micrometers in size. So common features of prokaryotic and eukaryotic cells. They both contain DNA. The genome is the total DNA within a cell. Genes are individual units of heredity, containing individual traits that are encoded to make a functional protein or RNA. Prokaryotes have smaller genomes than eukaryotes, meaning that prokaryotes have fewer genes compared to eukaryotic organisms. Both eukaryotes and prokaryotes have membranes. A cell membrane or plasma membrane separates cells from their environment. The plasma membrane is the only membrane that's found in prokaryotic cells. Eukaryotic cells have other membranes that are important for forming organelles. Lipid molecules are perfectly suited to form cell membranes. So for both prokaryotes and eukaryotes, the cell membrane consists of a double layer or bilayer of lipid molecules with a variety of proteins that are embedded in it. Some of these proteins embedded in the bilayer actually have a role in transporting specific substances across the membrane barrier in both directions. Both eukaryotic cells and prokaryotic cells have ribosomes. Ribosomes consist of RNA and proteins, which is also why they're called ribonucleoprotein particles. Ribosomes are the sites of protein synthesis in all living organisms. In eukaryotes, ribosomes are frequently bound to the endoplasmic reticulum, while in prokaryotes, ribosomes are usually found free in the cytosol. Prokaryotes have smaller ribosomes than eukaryotes. Both prokaryotes and eukaryotes contain cytoplasmic regions. The cytoplasm refers to the portion of the cell outside of the nucleus, whereas the cytosol refers to the aqueous portion of the cell that lies outside the membrane-bounded organelles. This slide shows the main structural features of prokaryotic cells. E. coli is the most well-studied prokaryotic cell, and so we'll often refer to E. coli in this class as a cellular model. On the left is an electron micrograph image of an E. coli cell. Prokaryotic cells contain a cell envelope which contains the plasma membrane and oftentimes a cell wall outside the cell membrane that serves as an extra layer of protection. While prokaryotes do not contain a membrane-bound nucleus, they have what's called a nucleoid region, which contains the genomic DNA in the form of a long circular DNA molecule. Many prokaryotic cells have specialized structures that protrude out of the cellular envelope, such as pili, which aids in surface adhesion, and flagella, which is involved in cell motility. The main difference between prokaryotic and eukaryotic cells is the existence of membrane-bounded organelles in eukaryotic cells. An organelle is a part of the cell that has a distinct and specific function and is surrounded by its own membrane within the cell. Examples of organelles include a nucleus with a nuclear membrane, respiratory organelles called mitochondria. These are structures in which energy-yielding oxidation reactions take place in eukaryotes. In prokaryotes, similar reactions occur on the plasma membrane. An internal membrane system called the secretory system, which includes organelles such as endoplasmic reticulum and Golgi. The presence of a nucleus in a eukaryotic cell is the defining organelle of eukaryotes. Structural features of nucleus include it is surrounded by a nuclear double membrane, usually called the nuclear envelope, pores in the nuclear membrane that allow the export of RNA into the cytoplasm, the nucleolus, which is a dark staining region or regions that are rich in RNA in the nucleus, and chromatin, which is an aggregate of DNA and protein. The chromosome is the most highly compact form of chromatin and is the largest level of chromatin structure. Most eukaryotic cells contain mitochondria. Like the nucleus, the mitochondria has a double membrane. The outer membrane has a fairly smooth surface, but the inner membrane exhibits many folds called mitochondrial cristae. The area between the inner membrane and the outer membrane is called the intermembrane space, and the space within the inner membrane is the mitochondria matrix. The mitochondria is the location of oxidation processes that yield energy for the cell. Most of the enzymes that are responsible for these reactions are embedded within the inner mitochondrial membrane or reside in the matrix. Mitochondrial DNA and ribosomes, which are similar to bacterial ribosomes, are found in the matrix, and some proteins are encoded and translated in the mitochondrial matrix, but actually most of them are encoded in the nucleus, translated in the cytosol, and imported into the mitochondria. The endoplasmic reticulum is a part of a continuous single membrane system throughout the cell. The membrane doubles back on itself, giving it the appearance of a double membrane. The endoplasmic reticulum, or ER, connects the cell membrane to the nuclear membrane. The ER can be rough or smooth. The rough ER, or RER, is studded with ribosomes that are bound to the membrane. These ribosomes, which can also be found free in the cytosol, are the sites of protein synthesis in all organisms. The smooth ER, or SER, does not have ribosomes bound to it. It is often involved in lipid synthesis. The Golgi apparatus is separate from the ER, but frequently found close to the smooth ER. The Golgi is a series of membranous sacs or vesicles that have the appearance of a flattened stack, so it sort of looks like a stack of pancakes. The Golgi functions in the sorting of proteins and secretions of proteins in the cell. It is also the site where sugars are linked to either cellular components, such as proteins. Vacuoles, or lysosomes, are acidic compartments for protein degradation and acts as sort of a stomach of the cell by digesting certain cellular components. Most things are delivered to vacuoles and lysosomes via the secretory pathway, but there's also a direct route from the cytoplasm that uses a cellular process called autophagy. Lysosomes are in animal cells and are very small and acidic. Vacuoles are in other eukaryotes, so these are large and moderately acidic and have additional roles such as maintaining cell structure by osmotic pressure and serving as sites of storage compartments. Other organelles include peroxisomes. So peroxisomes contain enzymes involved in the metabolism of hydrogen peroxide, which is toxic to the cell, and chloroplasts, which are endosymbiotic organelles that are only found in plants and some algae, and performs photosynthesis. Eukaryotic cells contain a cytoskeleton. The cytoskeleton is a network of filaments that pervade the cytosol. Major classes of cytoskeletal proteins include microtubules and actin filaments. The cytoskeleton is like a human skeleton in that it maintains the internal structure of the cell. It helps to move or keep organelles in place, such as mitochondria, which are attached to the actin and microtubule filaments. Plant cells, like bacteria, have cell walls external to the plasma membrane. A plant cell is mostly made of polysaccharide cellulose, which gives it its shape and protective strength. Cellulose is also a major component of wood, cotton, linen, and most types of paper. Plants contain chloroplasts, which are photosynthetic organelles, and are also found in algae. Animal cells have neither cell walls nor chloroplasts. To sum it up, both prokaryotes and eukaryotes have cell membranes and ribosomes, as well as genetic material. Prokaryotes do not have organelles such as nucleus, mitochondria, or endoplasmic reticulum. Eukaryotes contain organelles. In particular, they contain a nucleus, with chloroplasts existing as specialized organelles in plants and green algae.